Hello and welcome to this module on the new rapid amplification of cDNA ends, or RACE for short. In a previous module, we discussed the classic RACE, and in this module, we will be discussing the new RACE, which differs in various ways. So let's begin with an overview of new RACE. The first thing to recognize is that there are differences between classic RACE and new RACE. And in red, I highlighted the steps in new RACE that differ from the classic RACE. So for example, in the new RACE, you begin by dephosphorylating the degraded messenger RNA within your sample. In addition, you will be performing decapping of the full-length messenger RNA, and then you will be performing ligation of a synthetic RNA oligonucleotide, either to the 3' end or the 5' end, depending on whether you're performing 3' RACE or 5' RACE. I will discuss the need for dephosphorylation, decapping, and ligation, and why they're important as I discuss 3' RACE and 5' RACE. But for now, I just want you to understand that these in red are the major differences between the new RACE, or I should say these are the additions in new RACE as they differ from the classic RACE. And then after you conclude those steps of dephosphorylation, decapping, and ligation, the central concept in new RACE is essentially identical to that of classic RACE. You're going to be performing reverse transcription of your modified full-length messenger RNA. In this case, these are modified full-length messenger RNA because you have concluded those steps. And of course, reverse transcription is the process by which you create your cDNA. Afterwards, you will amplify the cDNA, and similarly to classic RACE, you will use 2 sets of amplification, whereby the second set utilizes nested primers. So let me begin with a discussion of the new 5' RACE. So as I said earlier, the new RACE differs from the classic RACE in the sense that you have additional steps that you will be performing to the RACE protocol. So in the case of the new 5' RACE, as is with the new 3' RACE, you'll be performing dephosphorylation, decapping, and ligation. The only difference between the new 5' RACE and the new 3' RACE will be here in the ligation step, as well as here in the reverse transcription step. And you can see that I highlighted the differences between the 5' RACE, the new 5' RACE that is, and the new 3' RACE in purple. I'll get into that in a bit. So the first step in new 5' RACE is dephosphorylation of degraded messenger RNA. So the idea here is that you have a sample with degraded messenger RNA as well as full-length messenger RNA. So your sample will inherently have some degraded messenger RNA, as you can see here. And what's important to recognize is that the degraded messenger RNA can self-ligate to one another. That is, the components of the messenger RNA fragments, or the messenger RNA fragments themselves, can ligate to one another in the ligation step. So to prevent that, we will perform dephosphorylation of the degraded messenger RNA using an enzyme called calf intestinal phosphatase, or CIP. Obviously, a phosphatase enzyme, by definition, is going to remove phosphate groups, or I should say it will catalyze the removal of phosphate groups. And the degraded messenger RNA will then become inert during ligation. In other words, they will be inactive during the ligation step and will not self-ligate. On the other hand, your full-length messenger RNA will be unaffected by the CIP enzyme. So your full-length messenger RNA will not be dephosphorylated by the CIP enzyme. It will only be your degraded messenger RNA, and that's exactly what we want. So after dephosphorylation, you will perform decapping of the full-length messenger RNA. So recall that in RNA editing, you have a pre-messenger RNA, and this pre-messenger RNA will undergo capping and polyadenylation, as well as alternative splicing. Here you can see the 5' cap in blue and the polyadenylation signal at the 3' end of the mature RNA. In decapping, you will utilize tobacco acid pyrophosphatase, or TAP, to catalyze that reaction of removing the 5' 7-methylguanosine cap. The result is that you'll have an active or phosphorylated 5' end. So what do I mean by that? If you take a look at this figure to the left, you can see that you have the 5' end of a messenger RNA. To the 5' end, you have a triphosphate connected with a 7-methylguanosine cap. It's called 7-methylguanosine cap because you have a methyl group attached to the 7th position on this guanosine. And importantly, you should understand that the tobacco acid pyrophosphatase will cleave your triphosphate at this location such that it releases a pyrophosphate and your 5' cap. What you are left with is an active phosphorylated 5' end. By an active phosphorylated 5' end, we mean that you have a single phosphate group here that is able to engage in ligation with an RNA oligonucleotide, as we will do in the next step. So the next step is ligating a synthetic RNA oligonucleotide to the 5' end. And this is one of the major differences between the new 5' race and new 3' race, in the sense that you are ligating a RNA oligonucleotide to the 5' end in 5' race, whereas in 3' race, you will be ligating the RNA oligonucleotide to the 3' end. In reality, and to be more specific, the enzyme that you will use for ligating the RNA oligonucleotide, which is T4 RNA ligase, will ligate the synthetic RNA oligonucleotide to both the 5' and the 3' end. It's just that your use of primers in subsequent steps will be the differential aspect between 3' race and 5' race and how they interact with the location of the added RNA, synthetic RNA oligonucleotide. After ligating the synthetic RNA oligonucleotide to the 5' end, you will perform the steps that we understand to be fundamental to race or classic race, and that is reverse transcription, amplification, and a second set of amplification using nested primers. So you will begin with reverse transcription of the modified full-length messenger RNA in order to create cDNA. Obviously, reverse transcription is the process by which you create cDNA from messenger RNA. And to perform the reverse transcription, you will utilize a gene-specific primer. After performing reverse transcription, you will amplify the cDNA through a first set of amplification using gene-specific primers and reverse complement primers pertaining to the ligated RNA oligonucleotide. In the second step of the amplification, or in the second set of amplification, you will use nested primers that also include the gene-specific primers as well as reverse complement primers pertaining to ligated RNA oligonucleotide, except in this case, these are nested primers, so they are more internal. So let's take a look at the process of new 5' race through this figure right here. So as I said earlier, you begin by dephosphorylating degraded messenger RNAs, and you will end up with full-length messenger RNAs that have not been dephosphorylated, as well as fragments of degraded messenger RNA that have been dephosphorylated. So in this case, we have a full-length messenger RNA, and what you will begin by doing is removing the 5' cap using tobacco acid pyrophosphatase. After you remove the 5' cap, this is the 5' cap basically, and you can see here that it has been removed, you have a messenger RNA without a 5' cap, meaning you have a messenger RNA with the pyrophosphate here as well as the 7-methylguanosine cap removed. You will then perform the ligation step of the synthetic RNA oligonucleotide to the 5' end. So you can see here that this right here is the RNA oligonucleotide, and it has been ligated to the 5' end of your full-length messenger RNA. After ligation of this oligonucleotide, you will perform reverse transcription of your modified messenger RNA using a reverse transcriptase. In this case, you are using a gene-specific primer to perform the reverse transcription. What I want to note here is that this primer that you're using for the reverse transcription differs from the primer that you're using for the reverse transcription in 3' race, or new 3' race to be more specific. So again, what I have in purple here are the differences between the 5' race and the 3' race. So in this case, in the new 5' race, you are using a gene-specific primer for the reverse transcription step. And after you perform the reverse transcription, you will have a cDNA. You'll have your first strand of cDNA. And obviously, this is a negative sense strand that you produce. I've discussed why that is in a previous module on the classic race. But to understand this better, you have to understand that messenger RNA is derived from the negative sense strand, meaning it's the negative sense strand that's used as a template to produce the messenger RNA. And when you're performing reverse transcription, you are performing the opposite of transcription, and thus you end up with the negative sense strand, which is the original strand of DNA, or it's the sequence of DNA that is virtually identical to the DNA that was used as a template, the antisense strand, the negative sense strand that was used as a template for the transcription and the production of messenger RNA. Here you can recognize that you have the reverse transcribed RNA oligonucleotide. So now you have a reverse transcribed RNA oligonucleotide. And what's important to recognize here is that this oligonucleotide will have sequences within it, and we will use the reverse complementarity of those sequences as primers in both sets of subsequent amplifications. So in the first set of amplification, you will utilize 2 primers. You'll utilize a gene-specific primer, which we'll call the GSP1, and you will utilize another primer called the NRC1, which is reverse complementary to the NRC1 sequence in your reverse transcribed RNA oligonucleotide. And here I want to point out a flaw in this figure in the sense that you have this primer labeled as NRC1, and you also have this oligonucleotide, which is the reverse transcribed RNA oligonucleotide. Here it's made of DNA since it's been reverse transcribed, also labeled as NRC1 and 2. What they're trying to tell you here is that this oligonucleotide sequence of the reverse transcribed RNA oligonucleotide contains sequences that pertain to the NRC1 and NRC2 primers, but in reality, the NRC1 primer is not identical to an NRC sequence within this oligonucleotide that has been added. It is, in fact, reverse complementary to it. So the NRC1 here is reverse complementary to a sequence here. And so you'll utilize the NRC1 to extend or amplify the first strand of cDNA going this way. And this produces a positive sense strand. And then you will be utilizing a gene-specific primer in order to extend and amplify the cDNA going this way, creating a negative sense strand again. Importantly, what I want to note here is that the NRC1 primer, or the primer labeled NRC1 here, is going to work on the first strand of cDNA. On the other hand, the GSP1 primer here is going to work on the extended sequence, or the sequence extended by the NRC1 primer, such that the GSP1 is going to anneal to this region. After you perform the first set of amplification, you will utilize another set of primers to perform the second set of amplifications. But in this case, those primers are more internal. They're called nested primers. So you can see here that the NRC2 primer used here is more internal, and the GSP2 primer here is also more internal relative to the GSP1 primer.